Yes.
Amazing.
Okay, great. Our next speaker is here. Pietro Tardelli Canedo is going to tell us a tale of two morphs.
So, hi everyone. First of all, this is not my usual conference look. I just had surgery last week, so hopefully this won't impact the talk. But today I'm going to be talking to you about Neopurcellia salmoni and the first report of male polymorphism in the harvestman suborder Cyphophthalmi, which is a work that I did as an undergraduate at Sarah Boyer's lab at Macalester College. If you aren't familiar with Cyphophthalmi, they're also known as mite harvestmen, or Cyphos for short, and they are a suborder of Opiliones. They originated 345 million years ago and are comprised of over 200 species, all of which range between 1 to 7 millimeters in length. And they are leaf litter scavengers that inhabit tropical and temperate forests of all their major land masses of continental origin. They have achieved this global distribution despite being dispersal limited organisms, meaning they don't tend to have species ranges of more than 10 kilometers, of tens of kilometers in any direction. And one of the places that you can find these organisms is in New Zealand, which is home to 3 endemic genera. Arachia and Racaia, which are here in blue and yellow, are found both on North and South Island and are comprised of more than 10 species each, as well as Neopurcellia, which is found only on South Island and is comprised of only a single species, making it a monotypic genus comprised of only Neopurcellia salmoni. It has very little overlap with other mite harvestmen, the exception being this northernmost population here, which it coexists with another Arachie, and it inhabits forests of high rainfall, which in the South Island is very convenient because there is this giant axial mountain range known as the Southern Alps that creates forests of high rainfall to the west and drier areas to the east, and these west forests are exactly where you find Neopurcellia. As I mentioned before, Cyphos don't tend to have species ranges of more than tens of kilometers in any direction, yet Neopurcellia has this linear transect of over 400 kilometers, which is highly unusual for these dispersal-limited organisms. And in literature, this has been brought up as a possibility for cryptic speciation, which is what we decided to investigate in this project. We decided to look at the phylogeographic structure of Neopurcellia to see whether or not it should be split into multiple species. If you are unfamiliar with the work that goes into Cyphos, this is what collection looks like. You might think that this is teeming with life, and so many of them are here, but this is in fact only one of them. So if you do macroarachnids, you should be grateful sometimes. So we obtained CO1 data from 58 specimens from 17 different localities, both from our own collections as well as from museums that were on loan to us. And we reconstructed their tree using maximum likelihood and ultrafast bootstrapping for nodal support. If you are unfamiliar with phylogeography, just to make sure we're all on the same page, if you look at this cladogram, there's really not any relationship between geographic proximity and evolutionary relatedness. But when you see individuals and populations with strong phylogeographic structure, you would expect to see something more like this bottom cladogram, in which evolutionary relatedness and the closeness of the geographic distribution is highly correlated. And that just happens to be exactly the case for Neopurcellia. There is a very strong correlation between the evolutionary relatedness and the geographic distribution of populations, in which monophyletic groups tend to correspond to adjacent geographic regions, as it is expected in these dispersal-limited organisms. So we retrieved 2 lineages, the northern lineage here, in the blue and purple, which has 8 populations, as well as the southern lineage, which is here in the yellow, orange, and red, which has 9 populations. And we can further divide Neopurcellia into 5 sublineages, which we are determining as closely related, geographically proximate populations, and that's the color coding that you see on the map, as well as reflected on the tree here. So we decided to look into their population genetics to have a better understanding of the diversity of those populations. We used the NASP to calculate their haplotypic diversity, which measures the uniqueness of a particular haplotype in a given population, and we retrieved the value of 0.962, which is very high considering that the values range between 0 and 1. And this indicates that the individuals are not really moving out of their populations, which is further evidenced by the lack of shared haplotypes. There are only 2 shared haplotypes among all 34 that we retrieved, and one of them is between those 3 yellow populations, and another haplotype between these 2 blue populations here. We also calculated the fixation index, or FST for short, which measures the proportion of the total generic variance contained in a subpopulation relative to the total genetic variance. A value of 0 would imply that the 2 populations are interbreeding freely, a lot of gene flow, and a value of 1, on the other hand, implies that all generic variation is explained by the population structure itself, and that the 2 populations do not share any type of genetic diversity. We ran this analysis with 3 different binning schemes, comparing northern versus southern lineage, as well as each of the phylogeographic sublineages, that are the color-coded ones, and not grouping the populations at all. And for all FSTs that we ran, we retrieved values that were greater than 0.8, sometimes much higher, which is indicative of a high population structure. So this minimum gene flow and high population structure are similar to what we usually expect to see in species. So could Neopurcellia be, in fact, 2 different species? So when we looked into published data for slower evolving nuclear loci to assess the possibility of substitutions that could be diagnostic of major lineages within the species, we found that for 18S, all sequences were identical. And for 28S, there were fewer than 10 mutations found in over 200 base pairs of data, each unique to a single individual. Therefore, this lack of fixed variation in slower evolving nuclear loci halted any impulse that we had to split Neopurcellia into 2 species based on that genetic data alone. Which is why we decided to turn to morphology to see if there was any kind of indication that Neopurcellia was in fact comprised of more than 1 species. If you're unfamiliar with Neopurcellia, they are identified by 2 of its most iconic features, so this pair of curling scopulae on their posterior tergite, as well as this very unique segmented 4th tarsus. For Cyphos, males are the ones who bear the informative features that diagnose them to the species level, and females tend to look morphologically consistent. And while we know a lot about these diagnostic features, very little is known about their dorsal anatomy, which is what we decided to look at for information. We found, looking at the SEM, that they are sexually dimorphic, as expected, both exhibiting this even granulation, and the main difference being this presence of this lobed posterior tergite on the male, as opposed to the female, which is smooth. What was very exciting, however, is that we found a second male morphotype. This one, on the other hand, wasn't all even granulated. It had those larger granules and the presence of pores in their tergites, which is very different from this other male, as you can see here. And this is such a striking feature that we can actually see this under the light microscopy. So you can see here, larger granules, pores, and here's another male with even granulation, and I guarantee you they're from the same species. I checked many times. So it seemed very clear to us. We have 2 morphotypes, 2 lineages, 2 species. Wrong. This couldn't be further away from the truth. None of the trees that we looked at supported the monophyletic distribution of the traits. There was no phylogenetic pattern. As you can see here, northern lineage has them, southern lineage has them, and there was also no geographic pattern. In fact, many of the populations that we looked at, 6 out of the 23 had both males morphotypes occurring at the same place at the same time. And that was independent of the data collection, the altitude, no real environmental factor that could explain it to us. And even for some of those populations that we only found 1 of the morphotypes, this could be due to the small sample size, as we only had 1 or 2 males for some of them. This seems to be a pretty clear case of male polymorphism, which is when males from the same species exhibit different morphologies. In harvestman, this has been observed in 2 suborders, Erpunoi and Laniatoris, and is often related to armory, protuberances or spines in their dorsal morphology, or discrete morphs with respect to the thickness of their pedipalps or chelicera. That is the case for Pentopsalis here, in which those 2 different appendages are corresponding to different fighting styles. A couple of studies have found a very strong correlation with sexual dimorphism, in which male polymorphism in harvestman has never occurred in a sexually monomorphic species. That is, a species where males and females look exactly the same. They also found that this feature has been gained independently multiple times throughout the harvestman tree of life, suggesting there is a degree of evolutionary flexibility to it. Oftentimes, they are associated with what we call alternative reproductive tactics, which is a mouthful, so we just call them ARTs. This is when a species exhibits different traits or behaviors that enhance an individual's reproductive fitness, and is often the outcome of a strong sexual selection.
Pietro, that's 10 minutes.
Cool. Then that's often associated with ornaments or weaponry. In Neopurcellia, this is actually the first case of male polymorphism ever described in the suborder Cyphophthalmi. That is far to my knowledge. If someone knows of any out there, please contact me. I'll be very curious. And we found that it's only present in adult males, not females, not juveniles, which leads to think that this is another example of ARTs. Cyphos rely mostly on chemoreception and olfaction for their sensory perception, so we think that this pores could be some sort of glandular opening with some sort of sexual role that can improve either mate choice or species recognition and thus increase the fitness of that male. However, it is not being selected completely, as 32% of the individuals that we looked at were non-paired of the males. So in conclusion, we did find a strong phylogeographic structure in Neopurcellia, in which populations have a strong geographic component to them, acting in a similar manner to those of species. But while the genetic data suggests the presence of two species, there is a lack of diagnostic features in slower evolving loci and a lack of morphological or behavioral data that stops us from splitting Neopurcellia into multiple species. We also found the first case of male polymorphism in Cyphos, and we think that this is associated with some sort of alternative reproductive tactic. But in order to confirm that, we need to carry out a survey of Cyphophthalmi to see how widespread this trait is in their evolutionary history, but also carry out some histological and behavioral studies to better understand the form and function of those different morphotypes. I'll be starting my PhD this fall, in which I'll be looking into some UCE data that hopefully will shed some light into some of the more genetic aspect of Neopurcellia, and maybe will be the answer to our question of whether Neopurcellia can be split into more species or not. I'd like to thank all these amazing people who made this work possible, as well as our funding sources, and I am happy to take any questions if we have time.
All right, thanks, Pietro. And I think we do have just 1 minute for questions. I'm taking a look at the chat, and we have a question from Chewy, who's asking, what frequency did the different morphotypes occur at within populations?
So there were, I don't have the number for the overall population, so there were 68% of them were males, and then 68% of them were paired, and 32 were non-paired, but I don't have the exact numbers for each individual population. And it's kind of hard to have a reliable number because some populations we had 20 males, and some populations we had 2, and the numbers varied greatly. So I wouldn't be able to give you a thorough answer. So the overall picture is that kind of 2/3 of them tend to be paired.
Okay, great. Great. Well, I will